The brain is our most important organ. It's the brain that makes us who we are. It's the brain that helps us enjoy life. It's the brain that makes us productive in society. But because the human brain is the most complex in the animal world, it's also the one whose development is the most complicated. And the complicated development of the brain with the billions of cells that have to end up in the right spots with the right connections, that is what makes the brain extremely sensitive to toxic chemicals. And this is something that we have only begun to realize very lately. What we have learned from neuroscience is that the human brain is so complex. It's the most complex in the animal world, and it's also the most efficient. But this requires that every second during early brain development, every second, 200 new brain cells are formed. Now, these brain cells then have to migrate. They have to move by themselves, find their way to their final position where they need to meet with other brain cells and interact and make connections. All of these processes are extremely complicated and very, very sensitive to adverse effects of chemicals. 1 out of 6 children has some form of neurodevelopmental deficit, and that can be a whole different issues like the serious ones like mental retardation, but it could also be a very slight, let's say, reading disability. And if you count all of this, it's a major problem. And they know that in the schools. The teachers know about it. And in fact, it's even bigger than that, because what I'm talking about and what we have found in our studies of mercury-exposed children and studies of children exposed to pesticides before they were born is that the most common problem is simply that the children function at a lesser level. They are a bit slower. They don't concentrate as well. They don't really remember so well. They can't find the words. They have difficulty coordinating their finger movements, for example. Problems like that. So in most cases, we can't see if one particular child has been damaged because of pesticides or lead or mercury or solvents or something else. We can't see it. What we can see is average shifts in distributions. So that if we look at a population of children, those who have been exposed before they were born to, say, pesticides or mercury, they function at a lower level, on average. And that means that the whole curve has been shifted to the left, and there are more kids with problems, and there are fewer kids who function at a very high level. I first got interested in this problem of brain toxicity when I was a medical student. My brother was the press secretary of the first Minister of the Environment, the Danish minister, and they were in Stockholm for the United Nations first environment conference in 1972. And I was watching television and saw a demonstration of patients from Minamata in Japan, the fishing village where a company had polluted the fish with mercury. And I saw victims, with their spastic movements, and, there was a mental retarded child, and it was tragic. It made such a great impression on me, and I wondered, why are we not learning about this in medical school? In medical school, they said, well, the placenta will protect the fetus, and, as long as poisoning symptoms are not present, there's not a problem. Wrong. They got this wrong, but it took me a couple of decades to figure out why exactly it was wrong. So in 1985, I got interested in mercury, and I discussed with my colleagues whether we could start some examinations of children and figure out if the mercury present in the oceans, in seafood, etc., if that could cause toxicity to brain development in people, not like the serious pollution in Japan, but just the everyday seafood. Now, that got started in 1985, 86, and we published our first report in 1998. And last year, in 2013, the United Nations approved a convention to prevent mercury pollution. And the United States was the first country that signed it. But listen, 2013, and the evidence that we produced was published in 1997, and the studies were started in 1985. There's a long hiatus there. This takes decades to get enough documentation to inspire decisions, especially international collaboration on prevention of pollution. In Minamata, people died from mercury poisoning, and scientists then conducted autopsies, and they wanted to learn what did they die from, what happened. And when they looked at the brain tissue, it looked like the adults had lesions in the brain in particular spots. And children who were poisoned, they had more widespread damage. But those who were poisoned before they were born, they had damage all over the brain. And the crucial observation was that their mothers were okay. The mothers were not poisoned, but the fetus in the womb was suffering irreversible poisoning, and many of them died. When we began to analyze cord blood, that is the blood that you draw from the umbilical cord at childbirth, we found that there were virtually hundreds of industrial chemicals and waste chemicals present in the cord blood, so that it's very clear that the placenta does not protect the fetus. So it's not an armor, it's more like a sieve. So just about all of the chemicals we were looking at managed to get through. And that means that the mother's exposure during pregnancy and chemicals that she may have accumulated in her body during her lifetime are shared with the fetus. Some of them, those that are fat soluble, some of the chemicals will also pass into human milk. And therefore, she will also share her chemical body burden with her child through breastfeeding. And so, from the beginning of life, we are all exposed to chemicals that some of them are, of course, present in the environment because we live on a chemical planet, so to speak, but many of them are simply a result of environmental pollution and the usage of toxic chemicals in consumer products and for other purposes. And the concern is that if we're exposed to chemicals like that from the very beginning of life, from the beginning of brain development, then maybe some of them can cause adverse effects. So this is the big scientific question. How do we identify these chemicals? How do we figure out which ones to control better and to regulate. So when we classify chemicals, the ones that are known to cause poisoning in humans from clinical studies, the total number right now is 214, and 100 are pesticides. So the largest group of chemicals are actually chemicals that were developed to cause toxicity, not to us, of course, but to pests of various kinds. And many of them were developed to target insects, in particular the nervous system of insects. And I don't want to embarrass you, but the chemistry inside the brain of a cockroach is not all that different from the chemistry inside your brain. So when the pesticides can damage the nervous system of various kinds of pests, we also need to be very, very cautious, because if humans are exposed to it, then it can possibly damage the human brain, in particular during the very sensitive early development. So when we study brain functions in children, we normally use neuropsychological tests. Sometimes we use what is called IQ tests, but we prefer to use clinical methods to try to understand how the brain is working. And so we can't use a test, let's say, to make a diagnosis of the individual child. But for example, in the Faroe Islands, where we studied Mercury, we can see that there are tendencies toward less optimal functioning. And we can see that in very simple neuropsychological tests, but we can also see it when we put electrodes on the skull of the children and record the speed of the electrical signals inside the brain. And we can see that the nerve cells are slower in transmitting signals inside the brain, the more mercury the child has been exposed to. We've also done sophisticated brain scans where areas of the brain that are in actual use will light up. And one of the tests that we did was that we asked the child to do this finger opposition test here. So, and normally when you do this, then this part of the brain, the motor cortex on the opposite side, will light up. And so it does in children without exposure to high levels of mercury. But we can see that kids who were unfortunate to have elevated mercury exposures, we can see that they used both sides of the brain in order to do this with their left hand. And that is clearly abnormal. It's like a maturation of the brain that hasn't completed as it should have. So we have several sets of evidence, like slower nerve conduction, the fact that the different brain regions are not communicating the most efficient way, and we can see that there are tests that reflect particular functions where the children have deficits. We have looked at all of the human studies, all of the epidemiology studies, where we ourselves or our colleagues have followed children from childbirth and through to school age and even longer, and assessed brain functions in regard to the exposure to toxic chemicals from early development. Now, all of that evidence, like mercury, has been accumulated over many, many years. It takes decades to do these studies. And so far, we've been able to identify 12 chemicals that we are certain can cause damage to the human brain. And of course, it's lead and mercury, and then there are a couple of other metals. There are some pesticides and some solvents and some other industrial chemicals like the brominated flame retardants and PCBs. Now, that dirty dozen, if you want, is a very short list. It's a tip of an iceberg that we need to control better, because at exposures that we're all exposed to today, we know that adverse effects on brain development can happen. Then there is an additional group of chemicals where we just don't know yet for sure if they can damage human brain development. But we know that they can damage brain development in rodents. We know that from experiments with rats and mice. And when I go through that list, then there are maybe as many as 1,000 chemicals that can damage brain development in laboratory animals. And most of them, the vast majority of them, have never been examined in regard to human brain development. We haven't had the time. And most of them are not regulated at all in regard to protecting us from exposures. There's an additional source of information, and that is the clinical poisoning data that is from people being exposed at accidents at work, or unfortunate suicidal attempts, or other clinical poisoning cases where we know from the symptoms of the victim that the chemical can reach the brain and cause neurological symptoms. That means that the brain is, once you get the chemical into the body, the chemical will pass through the blood-brain barrier, get into the brain, and act and cause adverse effects to the brain cells. We think that all of these very same chemicals will also pass the placenta, and since they can damage brain cells in the adult, we think that the developing brain would be even more vulnerable than the adult brain. And that's a list of about a little over 200 compounds. Again, these compounds are not regulated for the vast majority's sake, and we're all exposed to them to a greater or smaller extent. So we have a whole picture, a whole perspective of chemicals that we are quite convinced represent a hazard to human brain development. I would say we have the tools. Number one, we have animal tests. OECD has developed a protocol how to determine if a chemical can cause damage to brain development. The test is there. We can do it if we want to. In addition, colleagues who work on cell cultures have developed tests using stem cells from the brain. And stem cells are uniquely sensitive to chemicals. And you can actually test them in the laboratory, in a Petri dish, and determine which ones are problematic and which ones are not. And that's a whole lot faster to do than waiting to conduct an experiment in rodents. So we have the tools to do it. All we need to do is to make the decision. In the meantime, there's also been some controversies. First of all, there will always be scientists who say, well, this is not proven and there's still some doubt, and maybe we need to do some more research. So there will necessarily always be uncertainties. But in regard to mercury, as with lead, with pesticides, with many other industrial chemicals, there is also some corporations, some industry interests that will have benefit from raising doubt. And this has been true with mercury as well. And so when my studies were evaluated by the National Academy of Sciences and the United States Environmental Protection Agency, then some industry interests developed websites and posters and whole page advertisements saying that mercury was totally innocuous, and don't believe these scaremongers, and so they try to ridicule me, and they're perfectly welcome to do that, because I think that in the end people will understand that science speaks a soft language. We will never express ourselves as if we are 100% certain about something, because there are always uncertainties. But what we would like to do is to inspire prudent and responsible decision-making in society. So it's an issue we have to deal with here in America, but it's also an issue that is of international concern. I participated very recently in a workshop with the World Health Organization, and it was clear from the discussion that the colleagues from the World Health Organization and international colleagues in the field, that they share this ambition that we must protect the next generation's brains from chemicals that can harm brain development. And I understand that the World Health Organization is eager to take the lead, but obviously nothing will happen unless the member states, like America, like the EU, will support that. So over time, we have seen that the Centers for Disease Control and the World Health Organization have lowered the exposure limit for, let's say, lead or mercury as more information came out. And that's, of course, not because lead and mercury became more toxic with time. It's simply because we had underestimated how bad these chemicals were. And therefore, it was only when we finally got better evidence that we realized, we've done something wrong in the past, we better decrease the exposure limit. So with pesticides, it has been a similar experience, but more often, pesticides simply get banned after maybe 1 or 2 decades of use, because then it's found out that they were more toxic than intended. We recently did a study in Denmark of greenhouse workers, women who worked in greenhouses, and they were not spraying with pesticides, but they were exposed to them because they were peeling the dead leaves off the plants and touching plants that had been sprayed. Now, in Denmark, there is a very strict regulation in regard to pesticides. So the pesticides were short-acting, they were not persistent, they were what we call modern pesticides that were thought to be less toxic. And there's also legislation so that if a woman working with pesticides gets pregnant, then she is excused from work and she's being paid unemployment during the duration of the pregnancy after the pregnancy has been determined. Now, we looked at those women's children at school age. And we had a comparison group of kids of other employees from the same facilities not exposed to pesticides. And it turns out that children who very early in the gestation had been exposed to these modern, safe, as they would say, pesticides still had deficient functions in the brain. So that really scared me, that we're not doing enough to control these chemicals. They haven't been tested sufficiently in regard to toxicity to the brain. And then we also have to look at the global picture, because pesticides that are banned here in the United States or in Denmark or the EU or elsewhere, they're still being used in the developing world. We actually produce them and exploit them. And then populations who have much lower safety controls, less efficient safety controls, are exposed to them under circumstances that I'm quite certain will cause chemical brain drain in the next generation. And we have a responsibility, I think, to help those countries protect the populations against these chemicals and promote brain development in the next generation. So the status today is that, number one, the consumer can do a little bit, but basically we cannot ourselves make decisions, the necessary decisions, in order to protect ourselves and our children against chemical brain drainers, the chemicals that will damage the next generation's brains. We can't. So something needs to be done in regard to the way we use chemicals in society. So basically, policy decisions are needed. Now, in this country, we have the Toxic Substances Control Act from way back in the 1970s, and this legislation has called for revision for a long time, and it's still being debated. Unfortunately, that legislation is totally irrelevant in regard to the protection of the next generation's brains. It doesn't help at all. So we're not testing whether existing chemicals or new chemicals can affect brain development. We're not regulating chemicals that we already know can cause damage to brain development. And we're not sharing the information that we do have with consumers so that they can perhaps make at least some decisions on their own part, and perhaps also inspire to a more responsible way of using chemicals in society. So some change is needed, and I think it's an ethical, I'd say, requirement that we realize that we have acted irresponsibly. There's a whole generation of children who have been exposed to lead and whose brains are not functioning optimally because of the exposures that they had. And we're now realizing that the same has occurred with mercury, arsenic, toluene, certain pesticides, and several other compounds. And there may be an even larger number of chemicals that have already caused that damage. We should not continue allowing that to happen. I think, being at grandfather age, I think that it's irresponsible for grandchildren and the following generations. We have to make it a decision to change this way of acting. And I'm speaking here as not just as a scientist, but as a human being, because I think this is an issue where public health science has shown some very clear results. And of course there are always uncertainties, But the signals are so clear that I would say not acting is unethical and irresponsible.